Thank you, Peter.
Yes, thank you very much. It's a pleasure to be here tonight. Peter and I actually just returned from Barcelona last night from teaching a workshop, and we have some fresh material to present to you today that we're very excited about. So to begin, and also in the theme of this forum, connections, we'd like to start with a quote that we're very much appreciative by Buckminster Fuller, who, if you don't know, was actually a professor emeritus and world fellow in residence for the consortium of the University of Pennsylvania from 1972 to 1983. I'll just read this for you. He says, "About 1917, I decided that nature did not have separate, independently operating departments of physics, chemistry, biology, mathematics, ethics, etc. Nature did not call a department heads meeting when I threw a green apple into the pond, with the department heads having to make a decision about how to handle this biological encounter with chemistry's water and the unauthorized use of physics department's waves. Nature probably had only one department and only one coordinate omnirational mensuration system." Now, just to give some brief background into my own work, I'm interested in sort of multi-scalar investigations into behaviors and how they impact geometry, form, material, and even issues of fabrication as an interconnective loop. So here we can see smooth muscle cells interacting in their extracellular matrix environment. Now, that has impacted a body of work in textiles, both digitally and in the material realm, where I'm interested in the links between pattern or code and three-dimensional geometries, and again, ultimately, how they interact with issues of fabrication and material. And this is one example of one of those products. This was from a project called Body Blanket. And as we investigate this scale shift, I'm also interested in the reinsertion of the body. And this, we'll get into this more in depth. This is branching morphogenesis. And finally, looking at the scale of building. And so this scale shift is something that is very important in the context of our work and how we negotiate that. And one thing that we want to mention from the very beginning is that we're not interested in directly applying what we find in the biological models as a formal pursuit in architecture, but more so to look at relationships and how those transfer across that scale cascade. So just a brief intro into what is generative design and design computation, and more specifically how that is applied into both architecture and biomedicine in the context of our collaboration. Generative may be considered as bottom-up in the negotiation of form, function, and structure. It's process-based. Parametric, on the other hand, is oftentimes hierarchical, sharing both bottom-up and top-down investigations through associative and hierarchical relationships. Now, the third topic deals with optimization, which is much more goal-focused, and that is to say designing towards a specific form or function, context, or structure. And one example that has come within this in architecture is the production of what are called genetic algorithms for, say, building facades and how those facades may modulate light and heat gain more effectively. And then the fourth, design science, which we would like to sort of situate ourselves in, we're defining as the visualization and simulation of complex dynamic form and function in context. Now, this is actually the environment that I frequently find myself in amongst practitioners, researchers, academics, engaged in contemporary techniques, digital techniques, and the broach of the design of tools. And that may be found in the form of algorithms and what is called scripting or writing code. And just a few examples of what I mean by generative. This is a student project where they investigated the use of a fractal system and how that fractal system was then applied against multiple constraints that productively contaminated the process. In this sense, the brief called for a research facility, a new research facility for nanotech, the nanotech field, where they looked at the distribution of program. And when I say program, this means labs, wet and dry space, and so on, and how that might be effectively applied through the use of the fractal organization. And this was one mature example of their proposal. And just an example of what I mean by parametric in architecture, here we can see the design of a component at a local scale that then modulates across the whole. And in this project, they were interested in various sun path diagrams and how that component would perform against that existing context or set of constraints. And then finally, an example looking at optimization. This student project was interested in working with a material system called tensegrity and simulating that in a digital environment. And he wrote his own tools to do that, but again was optimizing for a particular set of material constraints. And one thing that I would like to mention, one of my mentors, it was mentioned earlier, who founded the Nonlinear Systems Organization, Cecil Baumond, is really considered to be a pioneer in this look between form and algorithm. And this is an image from a project that we collaborated on with the advanced geometry unit called HEDGE. All of this has come together in the context of our collaboration to be called matrix architecture, which essentially is looking at code, context, and perception as an interconnected loop. And more specifically defined as embedding material systems with biological relationships and behaviors to generate models that are at once natural and artificial. So this reciprocity between environment or context and how that specifies form, function, and structure is something that will continue to unfold throughout the course of this lecture. And just a quick example that may be familiar to you, someone that we are quite fond of in terms of defining matrix architecture, in our opinion, is Frei Otto. And this is an image of the Olympic grounds in Munich that was opened in 1972, where he started with a number of experiments looking at models found in nature, such as soap bubbles, to learn more about behaviors and how that might transfer through scalable material systems. And here you can see a detail of that particular project.
So Peter mentioned that, is this on? Okay, great. Peter mentioned that I'm a cell and molecular biologist. My research trajectory covers an array of systems, starting with lung vascular development in the fetus and newborn. Pulmonary hypertension is also a large interest of ours, looking at the remodeling of the adult vasculature. And that segues through to our studies in breast cancer and most recently in the last 4 years work in LabStudio. So there's a common theme that connects all these pieces within my lab, and this is the field of matrix biology. So cells don't merely exist with one another. They're surrounded by a complex protein network which instructs them, and I'll tell you a little bit more about that, what to do. It's an architecture of sorts and a code unto itself. So, as Jenny mentioned, it might not be obvious from her introduction how we may intersect within LabStudio, but my work predominantly is interested in how code and context, and that being the matrix, specifies form and function. So we work at multiple scales, from genes, code, cells, tissues, and ultimately the body. So I'm sure these types of images are relatively familiar to you. We're entering, or maybe even exiting, a genome-centric, stem cell-centric world where, give us your DNA and we'll build your body. Give us a stem cell and we'll build your tissue. Well, none of this is possible without context. DNA code doesn't operate without the proper context, and stem cells certainly don't differentiate if they're not provided with that context. And what is that context? We would suggest that, next slide, half the secret of how these systems operate is outside the cell, within this extracellular matrix. And you've all heard of this stuff, collagen, elastin, hyaluronic acids. They put it in face creams, and it does absolutely no use there. But within your own body, it's there as a structural support for cells within tissue. It lends flexibility to cells. But it's also informational. It's highly dynamic. It's a repository of biochemical and biophysical information, and I'll give you an example of that in a moment. And importantly, it physically couples the outside of the cell to the inside of the cell. So this is a section through a capillary. You can see the blood, red blood cells here, and it's surrounded to a large extent by this fibrillar network of collagen in this case. If we take these cells out of the body and put them into a tissue culture dish, they continue to make this matrix, which you can see in green. And I hope you can appreciate from here that there's a physical, at least, continuum from this matrix through this internal tensional network called the cytoskeleton, all the way to the nucleus where DNA is housed. So one can imagine that changes out here, even at a physical level, could impact what's going on out here, and vice versa. And of course, this doesn't operate without genes, and I'm a molecular biologist. I totally believe in genes, but certainly this area of biology is gaining interest in terms of how it controls gene expression. So the matrix is also a historical document. If you look at your hands, if you've been cut, you'll see a scar. That's collagen that's been built up. And we would argue that it's a master regulator of function. So I'm going to try and provide you with some information here about how this system operates. I was lucky enough to train with Mina Bissell at Lawrence Berkeley Lab during my first postdoc. And she really was one of the first people over 30 years ago to propose that tissue context could have an impact on gene expression through its effect on structure. We're exactly facing this criticism with our work, but occasionally things are lobbed at us in terms of what we're doing. But we take some comfort in this quote from Mina. She says here that she'll never forget the reception she got from a prominent scientist visiting LBL lab where she worked. She gave him a paper she just published on the genesis of cancer, and this was looking at the way in which tumors could be modulated by putting them into different environments. He took the paper and held it over the wastebasket and said, "What do you want me to do with it?" Then he dropped it in. And that was 20 years ago, and certainly in the last 15 years, the idea that phenotype or tissue structure can dominate over genotype, the constellation of genes that are expressed, is certainly more accepted. And I'll give you an example of that. What you're looking at here is a cross-section. Maybe I can show you with this. This isn't an exact representation, but if you take human mammary epithelial cells, put them into a three-dimensional matrix, they will undergo a morphogenetic process that makes them more or less look like the breast in vivo. If you put tumor cells, regardless of what type of mutations they have, into that same matrix, they completely disobey that surrounding context. And you can see in the cross-section there's quite a lot of order here. These polarized cells become highly disordered. However, if you trick these cells into thinking they're in a normal environment, and you can do this in this case with antibodies, even though they have these genetic mutations that lead to cancer, they revert back to a normal structure and function. And you can put these back into an animal and they remain non-malignant. So matrix specifies structure, and structure can control how genes behave, and based on that, we would like to know how that happens. Mina also proposed this model of dynamic reciprocity, and just going back to what I talked about earlier, that there's this continuum from the matrix through the cytoskeleton to genes being expressed on the nuclear matrix. So one can imagine, again, what happens out here can certainly affect what's happening here. Put this down. Oops, I'm just going to drop the thing. So how does a matrix do that? Next slide. We can skip this one. The common language that Jenny and I have developed across our fields, I think we started with thinking about things like material and geometry and form. Material certainly, or the materiality of the matrix, can affect how cells react to it. In this case, this is work from Dennis Disher in bioengineering. Here at Penn, Penn's become quite well known for this type of work, where he's taken an extracellular matrix and just changed the way it feels to the cell. So it's either soft or stiff. And if you take a stem cell and place it onto that matrix, the cell probes it, feels it, and decides whether it's going to turn into bone if it contacts a hard surface, or it'll remain non-differentiated on the soft surface. So in this case, materiality, the same matrix with a different compliance, with the same cells, can generate a completely different fate. Geometry is also very important. Again, I won't go into this into too much detail, but one can control the geometry that cells adopt by stamping matrix proteins onto non-adhesive substrates. So here we have a round stamp, and here's a square stamp. Put the same cell type on it and treat it in the same way with the same culture media, and the round cells differentiate into fat on the left and into bone on the right. So here we have, just by manipulating geometry, the same cells end up with a different fate. This is work from Magna Tadaseva-Chute, MD PhD student in my lab. Again, taking human mammary epithelial cells that give rise to cancer, put them into a 2D matrix, and they remain flat, poorly differentiated, and don't function. If you put them into a 3D matrix, however, they undergo this morphogenetic event where they become polarized. If you give these cells lactogenic hormones, they'll even secrete milk into central lumen. Next slide. So it may seem that we're coming from very different directions, but at the start of our collaboration, Jenny and I immediately saw some overlap. So the methodology and approaches that we've taken certainly allow us to at least start a conversation. We're both interested in investigating dynamic, complex systems, issues of nonlinearity and hierarchical relationships. And as I hope I've shown you, the importance of environmental context and reciprocity between cells and that context are certainly an interest of mine and Jenny's.
I'd also like to mention that a lot of my own background and a number of the studios and electives that are being taught within the School of Design here at Penn and also at other universities actually occupies a very niche area within architecture. So some of those key terms that I explained in the beginning, generative, parametric, optimization, digital techniques, and so on, I think lent a really productive starting point because it's a way of thinking through a design process that's systemic and relational as opposed to top-down and compositional. And so these shared techniques for making use of computation as well as the development of customized digital tools to visualize complex data sets at multiple scales and in multiple media. And the examples that you see here on the stage are just one of many. We make use of what is called a 3D printer, where we can actually 3D print a digital model. So how can data be seen, even heard, or held has been something that has been very important to us. This image that you see here is actually the first day of an elective that we now co-teach together. We've taught it for 3 years now in the graduate architecture program, where we actually house graduate architecture students with postdocs and fellows in the Jones Lab. The first time that we launched this was over a summer period when we officially launched LabStudio, which became a testing ground for the elective format. We have since begun collaborations with material scientists and mechanical engineers, namely Shu Yang, and this is Erica Sabin, who was one of those first graduate architecture students to work with us, and she ultimately developed a thesis that came out of that work and is now just about to pursue a PhD in cancer biology. Keep in mind, she does have a Master of Architecture degree as well. And this is just a set of images from one of our final reviews with a number of colleagues, Peter Davies, Annette Fierro, Sanford Quinter, Andrew Lucia, and so on, who have played a very active and dynamic role in terms of providing productive critique. And these are some of the models that stem from some of the projects that you can see here. So we've established 3 clear research trajectories, and those have been our focus for the last 3.5 years predominantly. And we'll begin with surface design and go through both the scientific background that grounds that work, as well as more design-focused projects that have stemmed from that trajectory.
So just in a little more detail, This is a cross-section through the human breast. There's this lining of epithelial cells, and you can see it sitting on this green stuff, which is matrix. Now, if you take those cells out, as I mentioned, and put them into two-dimensional tissue cognitive environments, which is where, unfortunately, most drugs are tested. Clearly, it's not what the breast looks like in vivo. If you put it back into the 3D matrix, however, you do undergo this wonderful process where you can recapitulate this structure. So we use this model to test the ability of genes and other matrix proteins to modify this structure, even leading to tumorigenesis. Next slide. So this just shows the time course. We can measure various things. It doesn't really matter what these are at this point, but starting over from day 1 through day 8, you can see these things enlarging in size. They even undergo a programmed cell death event around day 5, resulting in this clear void space, which I think you can see over on the right here. So to visualize this, we use many different techniques. These are optical slices of one of these structures. This is not in real time. This is just moving through that structure, visualizing different elements. So these were the primary data that we provided to one of the architects to start with in that first summer. Next slide.
For the architects in the audience, we would call these serial sections.
Right, exactly. So the protein that I am predominantly interested in lung vascular development as well as disease and breast cancer is a matrix protein called tenascin. And these details don't really matter. The only thing that is worth pointing out is that this protein is not expressed in the adult, but it's highly expressed around tissues that are on the move, including surrounding tumors. Next slide. So Agner looked at a number of patients, over 300 patients with breast cancer, and essentially was able to show that as breast cancer advances, the amount of tenascin surrounding that tumor increases. Now, is that just a bystander, or is it actually doing something to promote and feedback to advance this disease? So we can test that using those 3D models by adding purified tenascin protein into the 3D mix. Next slide. And we wanted to also then in parallel develop tools to see what the structural role of tenascin was in modifying these. So before I met Jenny, this was about as good as we could get. It's not bad, and we can quantify the effects of tenascin on this 3D structure. This is using a common program called MATLAB, and it allows us to reconstruct these elements. Next slide. And doing that, we were able to show that tenascin modified the surface condition of these normal structures. It also promoted cell proliferation, so these things were also growing. Now, the reason the structural details become important is, was this growth outward or inward, and does that even matter? Well, it certainly does. Luminal filling, so cells growing inward, is a very good indicator of a poor prognosis in breast cancer. So we really wanted to know, is tenascin advancing that outward or inward? And would that allow us to then propose a hypothesis that we could test in terms of what else tenascin was regulating? Next slide. And this is the type of thing we can come up with from just purely looking at structure. And Agner went on to show that tenascin could in fact activate a gene that has been linked to breast cancer, this protein called c-MET. And next slide. Still, though, if structure is the message, we weren't satisfied with the way in which we were approaching this. So this is where our first mapping project came in collaboration with Jenny and Wei within LabStudio.
So that very first summer, which involved Erica Savig and Wei Wang, this is some of the work he produced. Surprisingly, some of the first applications and products within our collaboration were on the scientific side. And this is just an initial mapping project where we took the data sets that were provided to us from the Jones lab and developed parametric models, digital models, from those cross-sections, if you remember those movies that you were looking at, and then approximated a smooth surface based on an existing digital model. We also work with a number of geometric filters as a way of looking at relationships, geometric relationships, material relationships, and so on in different ways. And this is what is called a Delaunay tessellation, and its geometric dual, the Voronoi. And together, we were able to see proximity information based on sites in a more profound way, or in a cruder way. And in conjunction with that, looking at the tessellation across the whole.
This is a very exciting moment for me when we're interested in how cells pack within a particular geometry or volume. So being able to take what the architects and designers had created and export it into an Excel sheet so we could get quantitative data was something I never expected that would happen in this type of collaboration.
And in the production of tools and correspondences, we were quite interested in how adjustments of the exterior surface or basement membrane would have deeper performances, structural performances, and how that feedback perhaps could give us new clues about organization on a material level, geometric level, and ultimately perhaps at an architectural level. And again, just a close-up view of some of those initial 3D prints. These models making use of the Voronoi, which is based on, again, a proximity factor and also cellular packing. Now, this project, which was led by Masako Murata, was quite successful in the fact that she brought a number of external constraints to the biological research. And she specifically brought an interest in deployable structures, which essentially involve three things. You have the structure itself, the mechanism, and how that mechanism is programmed. And so in this diagram, you can see a basic scissor mechanism and adjustments of parameters that allow it to perform and have variable outputs. So she thought perhaps she could learn from the biology to ultimately achieve variable performances and program the deployable systems in a variegated way. And so she started by looking at cell-to-cell junctions.
And focusing at a very different scale at a subroutine that occurs when cells do pack together to encompass that lumen that I described earlier. In particular, she was interested in the way that cells zipper together and pull on one another through their cytoskeleton, and she used that as the basis for at least the initial set of ground rules for these deployable structures. I think there's a movie next of that zippering process from Paul Noble. So these are two opposing cells. This is actually an injured fruit fly epidermis, and you can see this is the wound that's been created, and as the cells probe one another, starting at one end, they start to zipper together. So she was interested in the communication between these two surfaces and how they give rise to new form.
And so in our teaching, one of the first steps that the students take after they've been immersed within the scientific models through scientific papers and data sets is to begin to extract and abstract a set of relationships that then give rise to a set of simulations or the production of their own design tools. And these are just very, very simple diagrams showing how she then began to reprogram those scissor mechanisms. Brought, again, constraints coming from material. She worked with both wood and acrylic and eventually aluminum. And these are some of the initial models where you can see rendered by the hexagons those two cell surfaces and the interstitial zipper structure that she was interested in probing. And so ultimately these models performed in a variegated way. So a manipulation over here may have a distal effect, whereas in this diagram of the model you can see a much more holistic performance, but it's quite different on the ground here. And then she ultimately took this to a studio at the time that I was co-teaching with Cecil Baumond, where the parameters that originally came from the biological inquiry fell behind, but again there was a residue in the models themselves that then allowed her to deal with the topic of scale more effectively, or what I like to call productively contaminated the process. And in this case, this was a brief for a pavilion. And so she then began to consider site at a macro scale and how that would then elicit new performances in the pavilion itself. And then she finally brought this to a much more mature prototype or working prototype where we worked with aluminum. What is in the process is called water jet cut in terms of digital fabrication. And she also worked with an understructure of struts that would meet the ground based on these footings. And so again, this issue of scale and having to negotiate the constraints in a very rigorous way was manifested yet again through another set of constraints that came from fabrication directly.
I think that the larger models that you see are clearly the most obvious part of the collaboration, but behind that, underpinning this, are pretty powerful computational tools that we can use to pretest hypotheses before even doing the experiments. You often don't see that within these models. It's not obvious, but this sort of loop that occurs back from the design to the lab is something that we're very much focused on within our collaboration.
And so in the interest of continuing to probe issues of materiality and how we might embed biological behavior to ultimately achieve what we're defining as matrix architecture, which is creating models and working prototypes at this stage that are at once natural and artificial, was manifested in this project titled Ground Substance that stemmed from the surface design trajectory and was a part of CGRAPH in 2009, just this last August. And one of the questions that I had when we started, and this was also designed along with Andrew Lucia, who's a design and research associate in LabStudio, was to consider hybrid systems that would then be embedded with the biological behavior and alternative modes of fabrication. And more specifically, thinking about the use of ceramic as a material medium and taking advantage of its plasticity as it negotiated variegated behaviors. And this is just an image of some of the first direct printed ceramic parts coming out of a 3D printer. Now, of course, along with that comes a series of productive constraints. In this example coming from clay, where there was a lot of post-processing that had to be done, and these were some of the initial families of parts that came out of those first prototypes. We also worked with a second material, in this case, which is a starch-based or plaster-based material, which is much more accurate in terms of tolerance. And here you can see, I really like this slide, the same part, different materials. This one is ceramic, and this is this plaster starch-based material. And obviously with ceramic, we have to deal with firing and the reduction of or shrinkage of the clay body. So in a zero-tolerance model, that becomes quite a big issue. And these are some images of the final installation. And there were tertiary systems that ran through this. You can see with the cable threads. And we were very excited to see that this particular model or working prototype was being shown not only in design and arts venues, but ultimately was also included on the cover of the American Journal of Pathology in February.
That's great. So the second project, which we're going to describe tonight, looks at the way in which cells move within the setting of pulmonary hypertension. Now, if you don't know what that is, the next slide should help you along. Next slide. You can move on to the next one, Jenny. So I direct the Penn Center for Pulmonary Hypertension Research, and within that unit, we collect cells from patients with pulmonary hypertension as well as their normal counterparts. Pulmonary hypertension essentially is high blood pressure within the lung. It leads to these types of lesions, which you see here. This is a normal vessel. It has a nice large open lumen. These cells that have occupied this, together with this brown matrix, which we've stained, started off within the normal vessel wall, but they've crawled in there and grown within that space. So we're interested in determining how that happens at the molecular level, and what are the behaviors that drive this process. Next slide. Next slide. So we spent a number of years, probably the last 15 years, working on pathways that lead from the normal vessel structure to this occluded component, The patients that are diagnosed with this, on average, have a median survival age of about 2.5 years. So it's a pretty severe disease with no cure. And the drugs that are employed are fairly wimpy. They certainly relax the vessels, but ultimately the disease progresses. Next slide. So we're interested in trying to personalize therapy for this disease, and one would have to either know the full array of molecular programs that are operating, or could you focus just on behaviors that differ between patients, whether it was screening for a drug or a target. We're doing this work in collaboration with Scott Diamond, who's sitting in the audience at the Penn Center for Molecular Discovery. So I'm just going to show you a little bit of work on the molecular diagnosis and then work in LabStudio that's looking more at behavioral differences between individual patients. Next slide. So in terms of a visual, this makes the designers, what would you say? I don't know.
Very excited.
Yeah. But it's a difficult thing to read. Each one of these represents a different patient, and each of these clusters represent different proteins that each patient is making, and this is being deposited within their blood. We've devised a way to screen patients, and there are clusters that represent particular proteins that do link to subgroups of patients, but it's a very hard thing to read. If you're trying to do this rapidly, maybe looking at a process rather than a molecular panel would be an easier option, certainly. Next slide. So, in terms of gaining ideas of how one looks at behavioral signatures, there's a lot of tooling out there already within my field. In this case, we're looking at the way in which cells move in a tissue culture dish. You can measure this in space and time, by placing a cell, say, on a membrane and looking to see how fast it moves through that. You can trace how cells move within a dish. You can even look at as I mentioned, the environments and morphologies that the matrix can imbue on these cells in the setting of motility. And very few papers on this, but even looking at the local morphologies at a different scale, at the scale of maybe 1 or 2 microns, this cell is about 40 microns in diameter on average. That's something else we can do, but there's no method that encompasses all 3 of these parameters. So Erica Savig in the lab decided to use ice dancing as a basis for trying to understand the choreography of cells within a tissue culture dish.
Yeah, so Peter and I were very inspired by a student project that came out of an elective course, which at the time I was teaching again with Cecil Baumond to form an algorithm. And this particular project was looking at the space and structure of compulsory ice dancing. And so he developed a set of tools, digital tools, to analyze that. And what you see here are some of the constraints that he considered, the centroid of the body, the extent of the arms and legs. And so the compulsory dances, the choreographies of these specific dances, were inputted into Excel documents and were essentially gave rise to a set of algorithms or steps that were then mapped in a dynamic way through time and in space as a digital model. And these were some of the signatures of those specific dances. And at a later point, he then introduced not just one dancer, but 2 dancers. And what was very important, I think what excited both Peter and I about a possible trajectory, was the fact that it was also designed in context, albeit in a crude way, as you can see rendered by the rectangular box. But there was a reciprocity, a give and take between the code of the actual dance, the extents of the body, and how it interacted in context. And so one of the initial starting points, just make sure this is playing. Well, we'll just look at the image, was to do just that. And one of the problems that we faced was we didn't have a set of choreographies to begin with in terms of beginning to map the actual choreographies and movements or motility of cells. And so we had to develop a series of tools, and these are Erica's first takes at designing her own code, her own set of algorithms to extract those important parameters and behaviors from the cellular data sets.
So although we haven't performed this with different patient cells, we have tested as proof of principle differences in behavior in different microenvironments. So we place cells in one type of matrix versus another to see whether these tools can distinguish between those. So Erica developed a very nice edge tracing algorithm that could select cells and look at them through time. Next slide. This is the Z direction. A cell moving through time. It starts off on the dish as a spherical object. It spreads and then starts to move. And this just shows you that movement. Next slide.
And importantly, this allowed us to not only just look at discrete shape in terms of the figure of the cell at any given moment, but to look at its overall morphology.
And what this algorithm is doing is every 2 or 3 microns, it's noting a point on that perimeter and giving us that coordinate. We realized early on that we would have to have that fine level of detail because most cells from this particular vessel wall, if we put them into culture, we certainly can't see those differences if we look at the perimeter as a whole. So in order to appreciate that, Erica created filters that would then allow us to look at things like twisting behaviors and directionality. Next slide. And importantly, new ways of visualizing these complex data sets. This is looking at this native environment. This is the same protein, but it's been modified slightly. And immediately it becomes clear that these behaviors are radically different, even in the same cell type, but placed in a different microenvironment.
So what you're looking at on the vertical axis is time, and so each one of those cell edges is actually unraveled, so we're able to look at a landscape, if you will, of that overall morphology of dynamic behavior.
And a very exciting moment for me was when Erica analyzed the quantitative data and went back to the movies to see what was happening at particular moments that looked interesting. She came up with a list then of new behaviors, and this is from a non-cell biologist, this is from an architect to student, and discovering many behaviors that we have some idea as to how these are occurring, but with this complete objectivity and no bias as she came into the lab, she was able to create these types of catalogs that we're very excited to understand how these different forms are occurring, at least at the molecular level. And these occur differently between different environments at different frequencies. Some of them occur within the native and not denatured, and vice versa. And she went on then not only to look at behaviors at this scale, but she can quantify these behaviors and translating those behaviors into different visual signatures. Next slide, I think there's one more of that. Okay. There is a problem with looking at individual cells in that these populations are very heterogeneous and not all cells behave the same. So Andy Lucia in the lab has tackled that by looking at a field condition. He's been making movies of these cells and looking at the cell population as a whole and changing the pixel data into a different visual signature, in this case a color code. Is there another slide of this? There we go. So what you're looking at here is if a pixel moves to a larger extent than an adjacent pixel, it will become red. A medium amount of movement will be green. Blue is very little movement at all. So he can extract these data and essentially graph them or even convert them into frequency barcodes that will allow us to look at the field as a whole rather than individual cells.
Yeah, and just to add to that, by looking at a very simple parameter like pixel brightness, we're able to see the change in behavior and capture that through what is essentially the difference between one event and the next through working with the images directly.
Once again, a different visual, certainly for this field, but this is looking at the native condition. This is frequency and time. And the denatured condition. And immediately, of course, you can see through this tessellation that was superimposed upon these frequency maps that the cell is behaving very differently in each of those individual environments.
This is again making use of one of the previous geometric filters that I talked about, which is called a Delaunay tessellation. which again allows us to look at a field condition as opposed to what Erica was working on, which is isolating individual cells.
So for the last project, we'll move quickly through this. We've designated this networking, and it looks at the way in which cells within the lung communicate with one another to form those beautiful fractal networks that you just saw. So we understand somewhat the process of how vessels arise within the lung, and we've spent, again, a number of years dissecting the molecular pathways that lead to this vasculogenesis or angiogenesis, this capillary network formation from pre-existing vessels that we all rely upon during lung development. Next slide. We know that matrix-dependent vessel formation is important because if we remove the matrix, in this case in a genetic model, vessels simply don't form, and the airways which accompany those vessels are also poorly developed. This is an animal in which is making a normal matrix. Over here we've removed that matrix, and the vessels which are shown in brown here fail to develop. So we're interested in how cells communicate, what is the relationship between them and their matrix during this process, and specifically in the context of lung disease within the newborn. Next slide. Tumor vessels also rely on this networking behavior. They're very different from normal vessels. So how do we distinguish between these, and can we use some of the tools imported from design computation to distinguish these differences? So once again, we create systems within the tissue culture dish that recapitulates capillary formation. In this case, the system is overwhelming the matrix and is collapsing onto itself. If you push the next one. Is there another movie?
Yeah, it'll play in one moment.
Yeah. These are the same cells, should be the same cells, without the matrix, and you can see that they fail to undergo this network formation that you saw on the right. So the students are presented with these movies, they make these movies, and we ask them to study them and to tell us what they think they're seeing, and to derive very simple rule sets of relationships, either in the aberrant condition or in the networking scenario.
And so again, one of the first steps that we're faced with, and also what we demand of the students and the researchers, is to extract a set of rules and relationships, variables, conditions, that can then give rise to a set of simulations and new questions. And this particular project just started with 4 basic rule sets from initial population of cells being seated within an environment, how they are attracted, how that environment may change geometrically and in a material sense. And then finally, how do you begin to design a set of feedback loops that ultimately give rise to new questions both in science and on the architectural and design sides.
And initially, when we present these systems to the students, we flood them with the biological data. This is taken at the workshop from this week. And we allow them to study the entire process, or as I said, subdomains within that. Next slide.
I'll just mention one additional things to that. We often find that one of the crises that the students are faced with is that of the starting point. And we both stress that there is really no right or wrong starting point, but ultimately to make that initial move.
And the underpinnings of the macro work that you'll see lies within the lab at the level of simulations, which are shown here. This is work from Andy Lucia and Chris Lee, looking at the way in which cells can distort that matrix and distribute a force throughout the plane of the matrix. Next slide.
So these are just some simulations that are generated in a parametric modeling environment, where in this case you can see the context or environment rendered just by a simple set of points that are static, and then a component that is imbued with its own behavior as it modulates that existing environment. This is a more advanced simulation produced by Jonathan Asher, where in this case you can really see, again, the reciprocal nature of the context visualized as a tessellation, and how that impacts the overall networking behavior of these components. And this particular project by Shuni Feng and Joshua Fries started with some initial questions that had to do with connectivity and clustering, and they were particularly interested in addition to that, scale-free networks, or looking at points that had the highest degree of connectivity. And this is a sort of really helpful diagram that many of the students start with, which again is to crudely render the rules and relationships that they're extracting from the biological models. In this case, looking at how environment is initially modeled as a surface, then as a weave, and finally as a translational surface that's imbued with its own behavior and again modulates the overall networking performances. These are some diagrams from that investigation. And what was quite interesting for me in the context of this trajectory was that while they didn't start with this as an initial goal, they ultimately developed some pretty interesting tools for the negotiation of surfaces and how those surfaces might be subdivided. And they looked at that both as a digital model and also as a set of, or family of 3D prints. And you can see here a number of those. And surprisingly, the models with the smoother surface actually had the highest degree of connectivity as opposed to those that were much more coral-like. And this was pushed further in the context of an independent study where they began to question the possible architectural relevance of these models, where again the matrix or context or environment is rendered in a three-dimensional scaffold system, and the component was then had to negotiate that in various ways. So we, as Peter mentioned, we just returned from a very exciting workshop in Barcelona that was a part of the Smart Geometry group and was housed at the Institute for Advanced Architecture of Catalonia. And this is an image of one of the very first days of the workshop before it was populated with many, many, many models. It's kind of amazing what we produced in 4 days. And this is an image of our crew. And just a set of images, again, looking at various modes of working and presentation. One of the questions that we had when we started planning for the workshop had to do with this issue of communication and the development of reciprocity between context and form, function, structure, and how that was imbued in materials, and also how that was manifested with the use of digital fabrication. And what you see here are a set of parts, again 3D printed, but in this case not representing a model, but making use of them as actual building parts or components. And the students were actually immersed in a number of scientific readings before the workshop, and we launched what we called a node competition, where they had to design their own families of nodes and develop their own rule sets that then had to negotiate the existing constraints of the space itself. And the students came up with a really interesting scheme once we broke them up into two teams, which had two different sets of relationships and rule sets. One started on the ground with the highest degree of connectivity and stiffness. They had to work with three types of stiffness, from a hard, rigid connector, a mid connector, which was acrylic, and then a third, which was a floppy nylon braided tube. And then the other team had an interest in starting with 5 tensile points that were at the realm of the upper regions of the roof structure. And then where it became very interesting was how they had to meet. So coming full circle back to one of the case studies that I mentioned with Frei Otto, this notion of designing with force as a parameter and learning from the biological models in terms of communication and behavior between environment and form was something that we were interested in probing here. And what you see here is what's called a turnbuckle, which we were able to modulate as a parameter, a very specific parameter in the project itself. And so the student here is actually imbuing tension into a cable system that ran through the entire network. And you can see some of the curvature that's starting to come about as they move upwards from the ground. And then finally to the upper points of tension. I think we got a lot of funny looks through the course of the workshop from the other 9 clusters wondering what the heck we were doing. But one of sort of exciting moments for me, and I think also for Peter, was when one of the structural engineers came up and mentioned that it reminded him of the catenary structures produced by Gaudi to understand tensile and compressive systems as a set of experiments. You can see here those catenary models that ultimately were used to produce the Sagrada Familia.
Yeah.
And just a quote that Peter put in where he states, "The great book, always open, and we must make an effort to read, is that of nature, and other books are taken from him, her, and in them are the errors and misinterpretations of men, women."
The last 5 minutes.
And we'll just close with the final project where the image that was used on the connections brochure stemmed from. It was designed by Andrew Lucia, myself, Peter, and along with Annette Fierro. And this stemmed from the networking trajectory. And in this case, we were interested in a couple of additional constraints. One was had to do with the topic of scale and how we might reintroduce or embed the body within that, and also how we might begin to slow down the process of visualization. And in that slowness, could we perhaps see something new? And some of those initial simulations that you saw previously gave rise to this concept, which was to work with 5 slices in time as a template, and then secondly, to work with the material system that many of you are hopefully familiar with. That was that of the cable zip tie or plastic tie. And this ultimately ended up playing a very important didactic role in terms of working with a ready-made that could disseminate very complex information, all 75,000 of them, and ultimately probe this sort of crossover between science, art, design, architecture as an interface. And these are some images just showing how we actually produced the piece.
Yes, and each one of these represents a data point from that simulation of a cell tugging on its matrix and the vector created from that.
And the time frame that this took was over the course of a full summer. So what started out with very sophisticated digital simulations then met an analog model that was proved to be very slow. I was very happy about that. And we worked sheet by sheet. The color represents density shifts in the field, and the densest moments were connected with tubes, also composed of cable zip ties, to allow visitors to peer through these time-lapse cuts and ultimately meander through them and behind, not crawl on them, although we did see that happen a couple times. And it's currently housed at Arts Electronica in Linz. And we were very excited to see how it was displayed in context with 2 simulations, one looking at live MRI imaging and the other looking at the movement of a model through one's eyes. And so this coupling of very sophisticated techniques for visualization with ours, which was very analog, and ultimately giving rise to questions about information, empowerment of individuals, and how models are best fit to do that was very exciting for us. And ultimately,
yes, very satisfying to work with Jenny and to find our work being appreciated now in a broader audience. So you preempted science, but this is the cover from last month. Next slide. I think that's it. Just like to say a few thanks. Been very fortunate to have funding from a number of sources, especially early on, some risk-taking from the university itself and the AIA. We just got the Scram Foundation grant, and ITMAT, which is directed by Garrett Fitzgerald here, has also been very helpful together with the IME in providing student support.
And there, of course, been a long list of participants from students, postdocs and fellows in the Jones Lab, and other researchers involved, as well as a number of mentors and collaborators. And we'd also like to extend a very warm thank you to the Penn Humanities Forum for inviting us to give this lecture. It's very exciting for us to be invited to programs that believe in this crossover between disciplines and the shedding of pedagogical boundaries, because ultimately that's what we feel is most important as a working philosophy.
Thank you.